So since everybody's quiet, how about we get started? So welcome to GradSem. I'm Andre. I'm Vicky. I'm back. Yay! Thanks for everybody for volunteering. You guys were great. We were actually oversubscribed this semester, so yay to our wonderful graduate students. You're awesome sauce. Faculty have white sheets. People in my class are going to have colored sheets, different colors every week. Surprise, right? Just to keep you on your toes. And we're going to start off with a little announcement from chemistry.
Good afternoon, everyone. I'm IC2Y from chemistry. I'm also happy to work with Dr. Eric May from here in MCB. So I'm just inviting everyone to the upcoming UConn Chem MCB Symposium 2019. It will be on March 30, 2019 from 9 a.m. to 3 p.m. in Chemistry A120. Abstract submissions are still open. If you want to submit, you can go to tinyurl.com/UConnChemMCB2019. The poster, I think, is in the elevator if you forgot the link, or you can contact Dr. Nathan Alder or me if you want to submit your abstract. We are open for oral presentations for student speakers and poster presentations. And we have prizes, I think cash prizes, I believe, for the best poster presentation. So if you're interested, just contact me anytime or Dr. Nathan Alder. Thank you. Thank you, everyone.
Okay, so let's get started.
Yeah, so my name is Barrett. I'm a first-year student in MCB. It's my privilege to introduce David Barrett. David graduated from University of Massachusetts, Boston, and he graduated with a biochemistry degree and is now a third-year student in his Y-Tech lab studying drug discovery. The fun fact about David is that he has a terrific roommate who's kind, attractive, all around wonderful, and it's his cat named Foo. And so without further ado, David.
Thank you for the lovely introduction. You're a fine roommate too. So our lab studies a cytotoxic T lymphocyte leukemia-derived cell line called Pol-104 cells. And these are CD8 positive T cells that are important in the adaptive community for clearing the body of virally infected and cancerous cells. Their function can often be disrupted by mutations in a target cell, changes in the tumor environment, medications, or by chronic stimulation. And one of the primary functions of CD8 positive T cells is the release of these cytotoxic granules, which is what we study for the most part. And with this assay that we've developed, we can readily measure this release of cytotoxic granules with a simple, easy to perform, inexpensive assay that's very amenable to high throughput screening. We figured out a lot of ways to mess up the function of the immune system. There's a ton of immunosuppressive drugs, and they're useful for a lot of different things. Compounds in glucocorticoids and cytostatics are employed to treat autoimmune diseases, aid in preventing transplant rejection. But despite all of these immune-suppressing compounds that we found, there's very, very few immune-enhancing small molecules that are known. I've taken the time to include a list of them on the left, imiquimod and lenalidomide. Aside from these compounds and a few derivatives of them, that's pretty much all we have. And so there's this great need to develop compounds that either enhance the natural function of immune cells or rescue their aberrant function during illness or the treatment of some illness. Everybody loves movies. And so in this movie over here on the left, the green cells that you see marked with calcium are some target cells. The blue cells are CTLs, effector T cells, that are marked with VERA2 and lysotracker in red. And what you see is the CTLs binding to the target cell, forming this immune synapse, releasing their granules, and one of the components of those granules ends up perforating the cell, the calcium leaks out, and eventually the cell dies. Now this process is very tightly regulated, in which the T cell receptor recognizes foreign peptide in the MHC of the target cell. Whole signaling cascade occurs in the effector T cell, and eventually these granules are released at the immune synapse. These granules have two principal components: perforin, which acts to poke a hole in the cells, and granzyme, which triggers a caspase cascade and ultimately leads to apoptosis. The way we might end up measuring Grandal Exocytosis is remarkably simple, and we take advantage of this other protein that hitches on a ride with the granules. So this protein, LAMP1, when the granules end up fusing with the surface of the cell, ends up localizing on the surface of the cell in direct relation to the number of granules that were released. And so after the granules are released, we can add an anti-LAMP antibody that's fluorescent. And when we run this through a flow cytometer, we're able to pick up on an individual cell-by-cell basis how many granules were released by that cell. When we first started doing this assay, we primarily used chemical stimulants, DG, Babs-Gargan, and PMA, which is a 4-bol ester. And this was great because these chemical stimulants produced a very maximal physiological response. And so if you were looking for things that suppressed immune response, you had a ton of room to find those things. But when you wanted to look for something that enhanced immune response, you couldn't really find that if the cells had already released all of their granules. And so we ended up switching to a system of stimulation with anti-CD3 coated beads. And so these anti-CD3 beads stimulate the T cell receptor in much the same way that binding to a target cell would. And the remarkable thing about using these beads in flow cytometry is that you're able to pick them up, pick up cells that are just bound to these beads. And so in this first figure here, we have forward and side scatter, which are essentially measures of size and complexity. And this population right here is our cell population of spread 1. And when you add beads here, you undergo a shift in side scatter that you can pick up of cells bound to beads. And this enables us to look at cells that have not been traditionally stimulated and cells that have. And so if you look at this figure on the right here, for CD3 beads, which are stimulating beads, no bead population has a relatively low anti-LAMP fluorescence, whereas the bead-bound population has a relatively high LAMP fluorescence. If you compare this to the chemical stimulus up above, it's still far below that. So we have some headroom to work between this CD3-based stimulus and what we know to be a maximal physiological response. So we ended up conducting a screen on a library provided by NCI, their diversity set library, which consists of roughly 2,000 compounds. And if any of you are interested in it, it's available free from the NCI. They'll send it to you. They'll send you resupplies of individual compounds. I highly recommend checking out their libraries. And we do this on a plate-by-plate basis. You can kind of ignore these first 20 wells here on the x-axis. Those are mostly controls. And what you see here is well by well. In black dots are what the bead-bound population anti-LAMP response is, and white dots is the non-bead-bound population. And the reason why we do that is because we're primarily looking for compounds that enhance the activation. And so for the white dots here, if you saw increases in the white dot anti-LAMP response, these are compounds that are activating the cell. We're more interested in compounds that enhance the response we're getting from bead binding. So we look at 2 different time points here, 24 hours, 30 minutes. And we end up finding a number of hits here. I've included some dose response curves for some of the compounds that we end up finding in the screen. But after you have these high throughput screening hits, what do you do with them? There's 2 things that anyone conducting any screen should do. It's pretty universal here. Is that you want to confirm the hits in the same assay in hierarchy. Okay? You want to weed out false positives. And the next thing you want to do is perform some orthogonal assay. Something that's measuring roughly the same process, but measuring it in a different way. And the question we're really trying to ask here is, does the increased LAMP response that we see actually mean that more granules were released? And these next things we move on to do are more specific to the questions we're trying to ask about immune function and what these compounds have an effect on. And so we asked, is the compound affecting closely related cellular processes? Is it affecting other things the T cells do, like the release of cytokines or cell killing? Is its effect cell type specific? Does it affect B cells, helper T cells, macrophages? And then this part's really tricky here. What's the mechanism? Of all the compounds that we found as hits from this screen, pretty much all of them were active at 30 minutes. And this suggests to us that their primary mechanism of action is likely not to be affecting gene expression, and that they're probably directly affecting some signaling pathway. And so that's mostly what we're going to focus on in this talk here. But first, we need to do some confirmation. And so of the dozen or so hits that we ended up finding from this assay, 7 initial hits reconfirmed in hierarchy. And so you can see here, y-axis again is APC mean fluorescence, so that's the anti-lamp level. And for pretty much every figure I'm going to show you today, we're always going to be comparing to this CD3 control. There may be some other controls on there, but the CD3 one is pretty much what you want to focus on. Because all of these compound-treated conditions are also stimulated with CD3 beads. What we ended up finding is that compounds change the proportion of responding cells to non-responding cells. This is important here because cytotoxic T lymphocytes undergo what's called serial killing. And this means they'll bind to one cell, they'll release a few granules, they'll move on, bind to another cell, release a few granules. And if these few granules are sufficient to kill the cell, it doesn't make sense to release more granules. And it's encouraging to us that this is increasing the proportion of cells that are responding to a stimulus rather than increasing their magnitude of response. Because if we cause them to simply release all of their granules, the first cell they bind to, they're not going to be able to engage in a serial killing. So next we wanted to see how much granzyme they were actually releasing. And for this we used a multiplex bead-based assay for soluble analyte detection. There are a few companies that develop similar systems to this, but they all work on basically the same principles. You have a mixed population of beads, which can be identified in a few ways. They're all different sizes, and they also have different fluorescences. And so each of these beads is coated in an antibody to one of the analytes that you want to be examining. And so when you mix it with your solution, they bind the analyte. Then you add another set of detection antibodies that are biotinylated. Those also recognize the analyte that's bound to the bead already. And then you add streptavidin P, fluorescent streptavidin. Those bind to the biotinylated antibodies that are bound to the analyte that are bound to the bead. Long chain there. Well, what you end up with is bead populations that you can separate based on size. You see this is a 4-side scatter gate here. We can separate these into bead size 1 and bead size 2. And for this, I jump ahead a little bit. We're looking at one of the cytokine experiments that I do later, but the principle is basically the same. You can see the different bead populations based on their fluorescence in this channel, the APC channel. And so you can see there's some IL-2 beads, some IL-10 beads, some TNF-alpha beads. And when you just mix them with media that has no cytokines in it, their fluorescence in this PE channel is relatively low. But when you mix them with a high cytokine standard, their APC fluorescence remains the same. They're still identifiable in the same way, but their PE fluorescence increases relative to the concentration of the analyte in solution. And so for our first experiment with these beads, we simply wanted to look at granzyme. As you might remember from earlier, granzyme is one of these components of the granules that are released. And so we wanted to see when we stimulated these cells, they have this increased lamp fluorescence on the outside of them, are they actually releasing more granzyme in the solution? And what we found was that of the 7 hits that confirmed the lamp assay, only 2 of them significantly impacted the amount of granzyme that ended up in solution. And so we decided to go ahead with 3 of them. 2 of them were from the previous assay there that we confirmed with the granzyme assay. And those were the 11643 and 7420. We also decided to go ahead with 125095. Even though that didn't confirm in the granzyme assay, it was by far the strongest of all the hits in the initial lamp response assay. And in addition, it had a very clean dose response curve. So even though it wasn't increasing the amount of granzyme in solution, it was still doing something interesting, and we wanted to continue to investigate that. So next, we wanted to see what these compounds had an effect on for cytokine release from these cells. And what we found was that of the 3 compounds that we decided to move forward with, all of them affected cytokine release in some way or another. One of these compounds, 11643, increased the levels of IL-2, IL-10, and interferon gamma. You can see 11643 is this blue here. 125095, the one that didn't confirm in the granzyme assay, decreased the level of interferon gamma. And 7420 decreased the level of IL-10. Despite literature reporting fairly significant amounts of TNF alpha being released by Tau-104 cells, we weren't really able to detect that. And so there was no significant difference between our controls or the compound treatments. So when you start getting into signal transduction, things get really crazy and complicated. I included this figure here. You look at it, there's like 50 things going on, and all of these are the result of a single stimulus event, where the T cell receptor CD3 complex is activated, and you get a ton of things going on here. The important part is not all of the individual things going on here, because this is a gross oversimplification of what's actually going on in the cell. But we need to start somewhere. And so we figured, why don't we look at signaling pathways that are shared between certain cytokine production and granule release. And so we had a clue of a few of these things that were shared between these two processes. And our methodology here was, let's start with the simplest ones first. And so the first thing we decided to look at was calcium inflows. This is a time course-based assay where at 0 minutes, CD3 beads are added, and you read the level of calcium in the cell at 1 minute, 5 minutes, 10 minutes, and so on. And the way you read calcium levels in the cell is with flow 4. And what flow 4 does is it undergoes a shift in fluorescence when it binds the calcium in the cell. And you can see that as this FITSI signal on the y-axis. And what you should see is within 5 minutes or so, there should be a sharp increase in FITSI signal, a sharp increase in the amount of calcium that you see. That tends to taper off a bit or decrease in the few minutes following that, and then you tend to see a slow increase afterwards. And that's what we see. That's what our control looks like. That's great. But none of the compounds had any sort of significant effect here that we can see. Now, currently underway, we have some data in the works examining a few other members of the signaling pathways that we know to be shared, including PKC and ERK activation. Now, we'll be studying these in a relatively similar way, in which we examine them through phospho-specific antibodies. And for PKC, instead of looking at PKC directly, PKC phosphorylates a variety of targets in the cell that share this common amino acid sequence. And there are phospho-specific antibodies that can be used to measure the degree of phosphorylation of these substrates in aggregate. And so this gives you an estimated idea of the level of PKC activity by the degree of phosphorylation of the substrates of PKC. With ERK activation, we can be a little more direct. You can directly measure through antibodies that recognize 2 phosphorylated residues on ERK itself that are markers of its activation. You can also look at a few other things. Similar to PKC, you could look at ERK substrate phosphorylation, but you can also look at MEK phosphorylation, which activates ERK itself. And by looking at these 3 things, you can get some insights here. So if there's some sort of discrepancy between MEK phosphorylation, ERK phosphorylation, and ERK substrate phosphorylation, this might reveal changes in other proteins, phosphatases that act on any of these substrates. Moving into the future, we'd like to examine the effect of compound treatment on cell killing. As I explained earlier, because these cells undergo serial killing, it's entirely possible that even though we're increasing the amount of enzymes that are released, that we might not make them more effective at killing more cells. We're also going to be looking at the compound's effect on different immune cell types and processes. These include cytokine release in helper T cells, the Jurkat, and so on, also in B cells and macrophages. I'd like to thank my wonderful PI, Dr. Adams Wyfack, current lab members, Megan and Brandon, our undergrads, our former lab members. As you can see, this is kind of an outdated picture, so I had to take some Photoshop liberties to make it short. The NIH for funding, the Go MCB board for all your help with all the things we've done this semester, and my wonderful Kathy.
Excuse me, a question, possibly from a student?
take those 3 drugs that you work and try to compare their structure and look for what's similar between all of them, basically, and find the drug that would work even better?
So there's not a ton of similarities between their structures. We have been trying to do some structural analysis of them. In fact, one of these compounds, go back here a bit. This NSC-125095. So this is the compound that greatly increased lamp response, but we didn't see any change in granzyme level. And this moiety on here, this sulfonylfluoride attached to this benzene ring, is present in nearly every single chemical serine protease inhibitor that exists. And so granzyme is a serine protease. It stands within reason this might be functioning through some mechanism like that. It might be changing granzyme's structure enough that we can't actually pick it up with this bead antibody system. So we have some insights as to the structure, but as you can see, they're all pretty wacky, pretty dissimilar. Yeah.
So you're getting a difference in lamp response and granzyme response. Can you explain at the cell biological level what you think is happening there?
Yeah, so I guess I kind of just alluded to, with that one compound, the possible explanation is something's happening to the granzyme after it's secreted. It's possible that, so we do some of these experiments at a 24-hour time point. This in terms of drug treatment, it may be simply increasing the amount of lamp that's in the cell relative to the number of granules, and so you can see an increased response that way. But trying to tease out what that is pretty much what we're working on now. Thanks, guys.
Great. Maybe something a little different. I'm not a grad student. So for those of you who don't know me, I'm Rachel O'Neill. I'm faculty in MCB. And the reason I'm doing this introduction is we also have a group of people that are going to be speaking, and they're also not grad students. So back in 2003, MIT started a project based on their independent activities period. And this project was synthetic biology. And what they did in this one project was they modified a biological system to make cells blink. Out of that was born something called iGEMS, which is the International Genetically Engineered Machine Team. It's an international competition. Universities can have one team. And so as an example of the growth of this particular initiative, in 2004, so a year after MIT started this, there were 5 teams. In 2005, there were 13 teams. In 2011, UConn started and did the Jamboree in 2012. There were 170 teams. This past year, there were 365 teams, and they're from all over the world. And the idea behind these teams is the students lead the project. This is essentially a tech incubator, and the students are from majors all over the campus. We have B&E, MCB, Patho, EEB, nursing, German, and history. You name it, we've seen it. They have to come up with the idea themselves, right? They have to solve a world problem with this idea. So the very first year, the project was VitaBeer. They wanted to synthesize the production of vitamin D in beer. Surprisingly, VitaBread won that year for the same exact idea. So this wasn't too far off. They also have to participate in an inter-lab experiment. It's actually a bunch of different sites doing an experiment and taking measurements together. But they also have to do human resource outreach activities. So they have to develop activities that they work with high schools or the public. All of this is of their own idea. So what I do is basically make sure they have a place to do it. That's it. They have to raise their own money, which they do every year through massive fundraising efforts. They have to find their own team members. They have to design their own project. Everything they do has to use either registry products, which are from the Biological Registry of Modified Organisms, or they develop their own and submit them. And that's actually part of the competition, is that they're creating a synthetic molecule. This year they have 20 team members. About 12 of them are actively involved in the laboratory research, and about 4 to 6 of them are the summer people. The summer is when the actual project happens, so they take the year to develop the project ideas. They're looking for grad student advisors and faculty advisors that they can just bounce ideas off of. That we can say, yeah, you could do that, or no, you can't. That's not going to happen in a month or in 3 months. And then we just sort of let them go. So we give them the resources to be as successful as they want to be. Is there anything else I should mention? You guys have probably mentioned everything. They do a ton of stuff. I never cease to be amazed by the things that they do. But I'll introduce the 3 key players this year. This is Peter, Sharon, and Liz, who are going to talk about the project that just happened last fall. The team has won 3 bronze medals so far, is that right? 3 or 4? 3. Including on this project. They're starting a new project. So this is actually the project from last year. And fun fact, they're all avid runners. And they constantly fight over the color of apples. Leave it at that.
Okay, great. Thanks for the introduction, Dr. O'Neill. So yes, we are part of UConn iGEM. My name is Sricharan. I'm Peter.
I'm Liz.
And we're presenting our project from last year, which we call Barrett, biological Alkane synthesis to shuttle electron transport. So just a quick overview of what we're going to be presenting today. We'll take you through a little bit of background about iGEM, just like Dr. O'Neill talked about, as well as to our project. We'll talk about the actual core of our project, which is Alkane biosynthesis. We'll go on to future directions that we would take for this project if we were continuing it into the future. And we'll talk about our plans for the next project that's happening over the course of this semester into the summer and beyond. So this is a little bit of a review, but iGEM is a competition organized worldwide with this many people. We are standing right here. So we just presented in October the project that we're going to talk to you about today. And there are hundreds of teams around the world, all geographic regions that compete here. There are a lot of different aspects that go into an iGEM project. First, the core of it is the wet lab and dry lab, the actual research that's taking place, and the modeling that we try to do to make sure that our project is viable. We also have to present at our at the competition that happened in October, as well as create a wiki to showcase our actual research work. You can take a look at our website, as well as a poster presentation. iGEM also encourages a lot of collaboration between different teams. So this year we've been mentored by a few different teams at Boston University, MIT, and iGEM Toronto. Helped us with a lot of the technical work that we probably wouldn't be able to do if we were on our own, or just had the resources at the university. They've been working on this for years and they're pretty new to it. We also do a lot of work with the community, which we call human practices. Recently, and coming up in a couple of weeks, we do a strawberry DNA extraction module with middle schoolers. They get to smash up the strawberries and they really love it. We did a trivia night with the high schoolers at E.O. Smith. And we also have just recently held, a few months back, a science cafe featuring a speaker from MIT who talked about synthetic biology to a general audience made up of members of the community. So now we're going to transition into the actual core of our project, which was fuel production, right? So the place that we started was that a lot of the fuels that are produced today for cars, for transportation, for energy are often energy intensive and wasteful to produce. It starts at the extraction of these fuels, it continues with the transportation of the fuel, it continues with the refining of the fuels, and all of this contributes to a lot of energy loss. And we think that a sustainable future requires a different strategy. So currently, a lot of the fuel sources that we use are based on fossil fuels, and we hope that the future continues to some renewable resource, such as solar or wind. But our solution to this intermediate time period is to produce a biofuel that could kind of bridge the gap between these two phases and produce something that's a greener alternative to the current fuels that are mined today. And we thought a good place to start with this would be cars. Today, a lot of them use gasoline, and in the future, maybe some will use electricity or run on renewable resources. But for the existing infrastructure that exists today, cars that run on gasoline, maybe there's a greener alternative that can replace fossil fuels. So to start our project, we tried to talk to a lot of experts in the field about where the industry was today. So we talked to Dr. James Stewart, who's a professor who worked in the biofuel industry in the past. And he gave us some advice about where the biofuel industry is today and is going in the future. We also talked to Dr. Peter Gunther, who gave us a viewpoint on the economic analysis of how biofuels could work into the current energy sector. So what we learned is that a lot of the different types of biofuels that are produced today are derived from different types of feedstock, like cooking oils, corn, or algae. But these require intensive processing to take them from that step to the actual usable fuel. And there are often a lot of compatibility issues in trying to get these fuels into the gas tank of a car. A lot of processing has to go into that. We hope that our project, which involves direct production of short-chain alkanes, which is what gasoline is, can produce a fuel that requires minimal processing, but also is compatible with the current transportation needs of today. So just to review, our project is to engineer E. coli to produce short-chain alkane biofuels. We chose E. coli because we are a relatively new group, and working with E. coli as a taxi organism is relatively simple and will allow us to be pretty flexible with the types of plasmids that we're able to use. We're using short-chain alkane because that's a great drop-in replacement for the fuels of today. The way that we're going to do this is by engineering different parts, an actual sequence of enzymes that take glucose and transform it into a fuel. And we're trying, as I said, we're trying to modify certain pathways in literature to produce this fuel. But we also had a plan because a lot of the times the biosynthesis using microbes can be relatively inefficient. This isn't something that we were able to do during this project, but it informed some of the design decisions we made in making our pathway. So we'd like to talk to you about it a little bit. So there are some drawbacks to trying to synthesize a fuel or any other product using a microbial system like the E. coli that we're working with. First, you have to talk about where are we going to get the biomass to produce the actual fuel. We are starting with glucose molecules, which are pretty hard to procure and pretty expensive compared to something like some other source of biomass. So that's a problem. Also, microbial biosynthesis requires specific growth conditions for the microbes that you're working with. And often they have pretty low yields, as we were expecting with our project. So our solution to this through reading some of the other adjuvant projects that have been around and some other literature, was to use this interesting pathway that's found in the soil-dwelling organism called Shellonella undensis. So it is able to respire on metal in the soil by transferring electrons from its metabolism to the external environment. And we want to modify this pathway. Usually this pathway is used in microbial fuel cell. So the microbes are able to respire onto an anode and produce electricity through this respiration. We learned that the Shellonella can actually do this in reverse. And if you provide electricity to these organisms, they're able to incorporate them into their metabolism and often have a higher NADH amount of reducing equivalents in their metabolism. And we're hoping that this could help power the biosynthesis pathway that we are going to use in our project. So our idea for this would be to use a microbial electrosynthesis system, which took electricity provided to the microbes that we're using to produce our fuel to help them have more reducing equivalents to produce a higher output of fuel. And so we're hoping to power up our microbes. So to review, we're going to engineer E. coli to produce this biofuel, and our future direction would be to integrate the MTR pathway from Shellonella for bioelectrosynthetic capability. The one requirement for this is that to use this pathway, you need to have an anaerobic system, so that informs some of the design decisions we made in our pathway. Now I'm going to hand to Peter to talk about the actual pathway that we're using.
Thanks, Chair. So this is not the first time that an iGen team has worked with production of short-chain alkanes. Specifically, in 2011, the University of Washington created the Petrobrick, which is a 2-gene system using the fatty acid pathway to produce these alkanes. So we're looking to potentially improve upon this, make a more efficient pathway. So we consulted Dr. David Walker from MIT, who does a lot of work with synthetic biology and genetic engineering. He suggested that in addition to using the MES, microbial electrosynthesis system, that Sharon mentioned, we could also take advantage of the wealth of information about the metabolism of E. coli and engineer a potentially more efficient pathway and compare that to the Petribrick. So after doing a literature search, we settled on this alkane synthesis pathway outlined by Choi and Lee. However, there was just one problem with it as it applies to our use in the microbial electrosynthesis cell. 2 of the enzymes, FADD and SER1, are aerobic. And as Sharon mentioned, that's a problem because one of the most important parts of the MTR of the electrosynthesis pathway is the MTR complex, which is specifically anaerobic. So to address this problem, we did a literature search and found 2 equivalents of these enzymes. For FADD, FADK, and for SER1, PMT1231, and these are both anaerobic. So now we could use this in the microbial electrosynthesis system in the future. When we presented our potential pathway to Dr. Richard Parnas, UConn chemical engineering professor, he mentioned that we needed to consider all the outputs of the system, not just the alkanes, because there's no point in creating a pathway when the output you desire is dwarfed by other byproducts. So to address this, we introduced 2 knockouts of FADR, which is a global regulator that controls the production of fatty alcohols and fatty esters, and also FADE, which converts the fatty acyl-CoA to the 2-trans-anyl-CoA. Okay, now on to the enzymes. First we have TESA, which is endogenous to E. coli. With the L109P mutation we included, it increases the output of shorter chain alkanes, which are more similar to gasoline in the very end. And we successfully constructed this. As you can see here, we have the map, promoter, ribosome binding site, gene, double terminator. Next we have FADK. This is also endogenous to E. coli. Like I mentioned, it's the anaerobic replacement for FADD. And we extracted this from the E. coli genome and isolated using PCR. So here we have the map of the part we successfully cloned with the native ribosome binding site and the gene. Next we have ACR. This is endogenous to Clostridium acetibutylicum. We attempted to construct this using Gibson assembly of G blocks we ordered from IDT, but we weren't successful after a couple of tries, so future directions could be either reordering different G blocks with different overlaps to try Gibson assembly again, or using other methods such as Golden Gate assembly. Finally, we have PMT1231, native to Protococcus marinus. Remember, this was the anaerobic replacement for SER1, and catalytic activity requires 2 NADPH molecules for every alkane produced, and this is important going back to the use in the microbial electrosynthesis system, because using that it can produce more of these molecules. And we successfully constructed this using Gibson assembly with G blocks from IDT, and here's the map once again. So as you can see, there are 3 parts of this that we were not able to get to in the time we had over the summer, and that would be the 2 knockouts, which we're learning more about lambda reg recombination techniques, which is what we would have used to knock those out, and also, like I mentioned, constructing ACR using different assembly methods, such as Golden Gate. All right, I'm going to hand it off to Liz to talk about future directions.
Thanks, Peter. So originally we planned this out to be a 2-year project, and so in addition to what Peter just talked about, doing the knockouts and also finishing the ACR gene, once we actually had all of the parts in the bacteria, we would have had to characterize the parts and then test them in the actual MES to find out that we were actually getting the biofuels that we had hoped for. However, we kind of sat down and thought about it, both in terms of our time limits and our resources and level of knowledge, and we decided that we're not completely equipped to completely finish this problem, and also we have a whole group of new students who are really excited about being a part of iGEM. So we're going to start developing a new project so that they can both be a part of that project development phase, and also so that we can take everything that we've learned and potentially make a more feasible project for this next cycle. So like I said, right now we're in the project development phase, and so what this entails is really just asking ourselves, what's a real life problem that we can use synthetic biology to solve? So this doesn't really involve doing the actual science, but taking pre-existing science and looking for novel applications. It's really engineering club. And also another part of that that's important is finding a project that is feasible both in time and in our level of experience. So we're doing this by basically just reading a ton of literature, and we go through, make lists of all the ideas that we can come up with, and then we'll systematically go through and just narrow it down to our new project. So like I said, just that process of scaling a project, time, having fewer genes will help because then we'll have the time to completely make them and then characterize them and test our final project, which is a large part of what, I mean, obviously if you want to present your research, you need results to present. So in addition to the wet lab portion, we also do modeling as a part of one of the iGEM requirements. So right now we're looking at reaction kinetics, but that can take the form of a lot of different modeling types. We also are looking into learning new assembly techniques because that was a huge obstacle for us. Specifically, we want to learn about building assembly. So the other thing that we're doing right now is we're training our new cohort. The three of us are each running a training group, and we have 12 little new trainees, and they're super excited about iGEM. And so really what we're doing is just giving them the skills to get from the beginning to the end of an iGEM project, as much as we know that is. So going through PCR, transformations, primer design. We've started out with making plates and making media, and we're going to progress all the way to the end so that they have all the skills to this summer do an entire project. So this past summer was the first year that we actually had an entire team dedicated to this summer project, making it more of like a legitimate internship type thing than just a club that people are involved in. And so the students who are in training this semester will be able to apply, and we're hoping to get probably what Rachel said, 4 to 6. And so the goal right now is really to prepare them so that they can hit the ground running once the summer starts and do a really awesome project. So getting that project well planned out and also teaching them all the skills that they need. So, yeah, hi Rachel. So another thing that we're looking for right now is being undergraduates, there's a certain limit to our level of experience, especially because a lot of us don't directly come from MCB, which gives us a lot of really cool skills that we can contribute, but again, we just don't have the experience. And so we're looking for a panel of faculty and lots of grad students who we can go to for troubleshooting, planning experiments, just people to talk to and bounce ideas off of, because, I mean, you always need that, yeah. So really we're looking for people with any types of skill sets that are in the general area, whether that's MCB. I know we have computational biologists that would be really helpful for modeling. If anyone has any synthetic biology experience and is interested, if you think that anything is at all related, talk to us and we can definitely find some way that you can help if you would be interested. We're also looking for faculty who have experience both in synthetic biology but also in the topic of whatever project we decide to embark on. So we'd like to finish off by saying thank you to everyone who's helped us in the past year. I know Dr. O'Neill and some of your grad students have been a huge help, and also our club members and our sponsors. And if anyone is interested in becoming involved in you, everyone.
Any questions? Does anybody have any questions? Any questions?
Yeah?
So I have two questions. One, do these small chain alkanes, can they run a standard engine, or does it need some kind of modification like how biofuel, biodiesel does for those engines? And two, a lot of what you would have in your media could be really damaging to engines, especially like small engines that don't run very frequently, like water and other things. How would you go about purifying what you synthesize once you've done it?
So the actual production of alkanes from our engineered E. coli is going to be relatively small, quite small, on the order of grams per liter. So it's not going to be viable to actually take the media and put it into an actual vehicle. So the way we test for presence of alkanes would be probably to do some kind of separation based on the polarity of the alkanes, and then test it on and then test the composition of whatever extract we have. Yeah, so a lot of this is a proof of concept to show that alkanes can be produced by
Thanks.
So do you have to have a project start to finish every year? Is that like the iGEM mission?
Yeah. So some teams choose to do every other year, which obviously is a little bit more ideal because you have more time. But the tricky part about that is if you do it every other year, then when new people come in, there's less for them to be involved in. It's kind of in the middle of things. So we want to be constantly recruiting. So at the moment, we're on a yearly schedule, but that is something we've considered before.
That is pretty typical for, or the yearly cycle is typical. Most teams do that.
So it wouldn't be okay if you presented an update to a project that was, you were working on year before?
You can do that.
Oh, you could? Okay, that might be a good idea.
How